molecular biology chapter, that is a DNA as a genetic material. A DNA is found to be a genetic material is not an easy process. There are 3 experiments which is going to lay a foundation to find a DNA as a genetic material. Before that, there was a confusion that whether amino acid, proteins, RNAs, DNA or a carbohydrate can be a genetic material. After these 3 experiments, which provides the evidence to know DNA as a genetic material. Among this, the first experiment is going to be a Griffth experiment, which is also known as transformation experiment, because it is going to be made, this experiment is going to made evidence of transformation through this experiment. It was known to be a Griffth experiment because it was proposed by Griffth. It comes under the transformation principle because a bacterial transformation is going to provide a evidence for this experiment. So as we have said that bacterial transformation is going to be a main principle in this Griffth experiment, therefore a pneumonia bacteria is going to be used for the transformation principle in the Griffth experiment. A pneumonia bacteria is going to cause a pneumonia. This is going to be a main principle. A bacteria chosen by Griffth is going to be a pneumonia bacteria. This pneumonia bacteria itself has 2 strains. They are rough strains and smooth strains. Where rough strains are found to be non-pathogenic, but smooth strains are found to be pathogenic. Rough strains are non-pathogenic because it is going to be rough because it lacks capsule. A capsule is going to provide a protective layer for the pneumonia bacteria in smooth strain. This smooth strain bacteria is going to be pathogenic because of this outer layer, that is capsule, which is made up of a polysaccharide. This capsule is going to provide a pathogenic and virulent factor which can't be killed by our immune system. Therefore, it is pathogenic. But the rough strain lacks the capsule, so it can be killed by our immune system. Therefore, rough strains are found to be non-pathogenic and avirulent. But a smooth strain are found to be pathogenic and virulent because of its polysaccharide capsule, which is going to provide a protective layer for the smooth strain. As the smooth strains also consists of types which are S1, S2, S3, and rough strains are R1, R2 and R3 types. In this, a main principle is going to be rough strain and smooth strain has different antigens. Antigens are going to be found on the outer layer. Among the rough and smooth strain, both has different antigens. But a smooth strain can convert into rough strain by mutation. A mutation here we discussed is going to be heat. A heat is going to provide a mutation in smooth strain to convert into a rough strain. When it was heated off, when the microorganism, smooth strain microorganism are going to be heated off, it is going to break off its capsule and converted it into a rough strain. Therefore, it will undergo into a rough strain, it becomes avirulent. From virulent, it can be converted into avirulent. This is going to be a main principle behind it. Now, we are going to see how these rough strain bacteria and smooth strain bacteria are going to be employed by Griffth to undergo Griffth experiment or transformation principle, which is going to lay a major foundation to identify DNA as a genetic material. Here we can find in the rough strain, it is going to be rough outside, while the smooth strain there is going to be a layer, protective layer, which is made up of polysaccharide coating is going to be there, which is known to be capsule is present. But in the rough strain there is no capsule, therefore it is going to be avirulent, and smooth strain is going to be virulent because of this capsule presence. This virulence causes pathogenic action, which causes pneumonia, which may also lead to death. But the avirulent bacteria, that is O strain, of which may not causes the pneumonia. This is going to be a main principle behind it. So we are going to see the procedure step by step of Griffth experiment. At first, Frederick Griffth took two strains. As we have already saw, they are rough strain and smooth strain. Therefore, at first, Frederick Griffth injected rough strain bacteria into the mice. As we already said, that rough strain bacteria are going to be avirulent. Therefore, doesn't causes any pathogenicity. It does not causes pneumonia. Therefore, this avirulent, the mice is live and there is no illness behind it. When a smooth strain bacteria is going to be injected into the mice, it was observed that a mice suffered by pneumonia and died, because the smooth strain bacteria is going to be virulent due to its presence of polysaccharide layer, that is capsule. Therefore, it was attacked by pneumonia and died. He done these two steps as the base. After that, he done another step. As we have said that smooth strain bacteria can be converted into rough strain bacteria by mutation, which is going to be heat, which causes a mutation. So, a heat killed S3 bacteria. When the S3 bacteria is heat killed, it becomes rough strain, which has a characteristics of rough strain, which means these heat killed smooth strain bacteria are going to be avirulent and doesn't causes any pathogenicity. Therefore, this heat killed S3 bacteria is injected into mice. It does not suffer by pneumonia. It was live. But when the mice is injected with the mixture of living avirulent R2 bacteria and heat killed S3 bacteria, these two strains are going to be mixture of, that means the rough strain, which is avirulent, and smooth strain, which is going to be heat killed, which means it also mutated to rough strain, which is going to be avirulent. When these mixture are going to be injected into the mice, there was an unexpected sign. Expected sign is going to be a live mice. But when this mixture was injected into the mice, it causes a pneumonia and it leads to mortality of the mice. This is going to be a base for the transformation principle. Here, it was found that the blood of mice is going to consist of both rough and S strain. That means R strain and S strain, both were present. Which means that S strain is not heat killed in its blood. So here we can find that these S strain bacteria, though it was mutated, it has DNA. This DNA is going to be transformed into this rough strain bacteria, which causes it to behave like a smooth strain bacteria, which causes pneumonia. Therefore, Griffth concludes that presence of heat killed S3 bacteria caused the transformation into the R3. Therefore, though it was heat killed, it has DNA, which has a characteristics of virulence. So, it passes its DNA to rough strain bacteria. So, when it passes its rough strain bacteria, it causes it to behave as causing the pneumonia to become virulent. Therefore, here we can find the transformation principle. That means the DNA is going to be transformed from one bacteria to another bacteria. So, the S3 bacteria is going to, though it is heat killed, it has DNA which is transformed into the rough strain bacteria, which makes it to restore the capsule formation, which causes it to become a virulent and causes pneumonia. Therefore, the mutation lost by S3 bacteria is going to be restored here. Therefore, Griffth concludes this, there is a transformation to be taken place between rough strain bacteria and S strain bacteria, which means the S strain bacteria is going to provide its DNA to rough strain. Therefore, there is a transformation is going to be taken place, but Griffth does not conclude that this transformation is going to be a DNA or a protein or an amino acid. He doesn't conclude that, but he proved that there is one thing which is going to be transformed, which causes it to carry the characteristics. So the character is passed from one generation to another generation. Here a one bacteria to another bacteria, a characteristic is transformed by one biomolecule. Therefore, it is known as Griffth effect. So here we can see the Griffth experiment overall view. Overall view of Griffth experiment. According to Frederick Griffth, at base as S strain, R strain, which is going to cause the pneumonia. By S strain only causes pneumonia, but R strain not, because it is avirulent. When it was injected into mice, a S strain causes pneumonia, so it dies. But the O strain does not cause pneumonia because it is avirulent, which lacks capsule. Therefore, the mouse lives. But when the S strain bacteria is going to be heated, it causes it to lose the characteristics of S strain. Therefore, the mice live. Mice lives. There is no death to be occurs, and no pathogenicity of pneumonia is going to be taken place due to the loss of characteristics of the S strain. But when the heat killed S strain and R strain, live R strain are going to be injected, the expected result is going to be mice live. But when it was injected, it causes the mice to die due to pneumonia. When the underlying reason was examined, it was found that the S strain bacteria, though it was heat killed, it has DNA which has a characteristic to cause pneumonia, is going to be transferred to R strain, which can be converted into S strain bacteria, which causes the pneumonia, virulence, which leads to death of the mice. Therefore, here Griffth concluded that there is going to be a transformation occurs from one cell to another cell. Here the cell is going to be bacteria. Like this, every person is also going to transform one thing from one generation to another generation. Here the transformation is only concluded by Griffth. It was not confirmed that whether it was DNA, RNA, or a carbohydrate or a protein.